Good morning.
Good morning.
Am I audible?
Good morning, sir. Yes, sir, you're
Good
audible.
morning. First years. I guess most of them are here. Let me just tell you, we have a guest lecture today for you people as a part of a conference. The topic is much related for you. It's purely on microbiology. It will help you. So I have asked you people to come over here instead of our class. So be here for the next 1 hour. The next 1 hour talk will be for the entire conference, which will also benefit you. This is for your information. Now we'll start the conference. Muthu Kumaran. Muthu. Muthu. Muthu.
Sir, I can only starting, sir.
Nada.
நான் starting.
நீ ஸ்டார்ட் பண்றியா? சரிப்பா. ஸோ கெஸ்ட்டை இன்வைட் பண்ணிடு. நான் அதுக்கப்புறமா பேசுறேன். நீ ஸ்டார்ட் பண்ணிடு.
Thanks.
ஓகே.
Very good morning to everyone presented here for Day 2 National Conference of Biodrasillence VT'21. I am Punraj from Department of Biotech, VT'21. I am pleasure to be given a wonderful opportunity to invite our estimate chief guest, Dr. M. Pavitra. She has completed her MSc in Applied Microbiology and also later on she pursued her PhD from VIT of return-based composting sustainable agriculture, and the main project like therapeutic potential and molecular docking studies of seed extract against bacterial food pathogen. For this project, she received an Best Article Award for VICAN 2020. She has interested in various areas of biotech like clinical microbiology, immunology, soil microbiology, general microbiology, and molecular biology. Also, she published many journals and few more manuscripts are under preparation. And now she is working in BG Vaishnava College as assistant professor of Department of Biotechnology. And I would like to Ms. Dr. M. Pavitra to take over the session.
Before Pavitra ma'am takes over the session, I just wanted to add few words to what Punraj has said. So what Punraj was given is the clear professional perspective of Dr. M. Pavitra. In addition to that, I was very much touched with the way Dr. M. Pavitra was there when she was doing her PhD work. It was being seen that it was nearly 6 years of tenure. She has done her PhD in VIT Vellore, in addition to her BSc and MSc, which was also in Vellore, VIT. So during the tenure, we can see that the lab is totally dependent on her. Each and every aspect will be taken care by Dr. M. Pavitra. I was there in the lab. I used to work there. And the most important fact that has impressed many of us is the way she handles the microbiology lab and microbiology perspective of laboratory techniques. Because certain techniques needs hands-on experience. That experience don't come for everyone. And it takes years to learn it. I could see an expertise hand in microbiology when I see her working on microbiology techniques. So I wish our students gain some information from her through this lecture talk, in addition to the specified lecture which has been planned today. Now I would like to welcome Dr. M. Pavitra, well-wisher of mine for delivering her lecture today.
Thank you, Dr. Sai, and thank you for the person who gave a nice introduction too. So, good morning, one and all. I thank you for having me here. I thank the Sai Ramesh for organizing this national conference named Biodrasillence VT'21. To begin with, the food you eat can be either the safest and most powerful form of medicine or the slowest form of poison. So, food can be anything. It can be a powerful medicine or a slow poison. So, this presentation is about the slow poison. I mean, the threats of food infection. Let me share the presentation.
Archak, sharing option இது பண்ணுங்க. ஷேர் கார்டே ஒரு சின்ன ஆரோ இருக்கும், அங்க போயிட்டு மல்டிபிள் ஷேரிங் கொடுக்கும்.
Now you can share, ma'am.
Yeah, it's shared all. So நீங்க ஸ்டார்ட் பண்ணுங்க.
Okay. I think the screen is visible, right?
Yeah. Yes.
Thank you.
All the best.
Thank you.
ஒண்ணும் இல்ல சார். ஏன் விளையாடிட்டு இருக்காங்க?
இது போக மாட்டேங்குதுப்பா. Sorry, just a minute. Okay. Sorry for the inconvenience. And we are beginning with the EHEC threats and challenges. Are you hearing me? Am I audible?
Yes, ma'am.
Yes, ma'am.
Yeah, thank you. So EHEC threats and challenges. So what is EHEC? Can you tell me? Can anybody tell me? Okay, so overview of this presentation is we'll have a small introduction on this EHEC, then the threats. So in what way it troubles the environment. Then we have a report, a case study of this particular pathogen, okay, this particular group of E. coli. Next, the conclusion of this case study. Then it will end with the challenges, in what way it is challenged. What are all the challenges faced by the environment and faced by the scientists and the people by the EHEC. Okay, so we'll have a small introduction on E. coli. So E. coli is a gram-negative facultative anaerobe, and it is rod-shaped. It is a bacteria, rod-shaped bacteria, and it is found in the lower intestine of endotherms. Endotherms means warm-blooded animals. Okay, so it was discovered by Theodor Escherichia in the year 1885 from the feces of the healthy individuals. So it was in the colon. That's why it was excreted in the feces. That is why it gained the name coli, as it was present in the colon of the human beings and the ruminants, warm-blooded animals, etc. And by the name Escherichia, who found this, who discovered this, its name was E. coli, that is Escherichia coli. Before that, he named it as Bacterium coli, but to pay respect to Theodor Escherichia, who discovered this, it was named as Escherichia coli. So it is an autochthonous member of the gastrointestinal tract of humans and warm-blooded animals. So with or without genetic determinants of pathogenicity. So sometimes it may have the pathogenicity, sometimes it will remain as a commensal, it will be very silent, and we'll mistake it as a very good organism, but sometimes it will reveal its pathogenicity and it will cause diseases in human beings. Okay. So next is the pathogenic E. coli are responsible for three major types of clinical infections. So what are these three major types of clinical infections? So one is enteric and diarrheal diseases. The next, urinary tract infections. Next is the sepsis and meningitis. So all very serious infections are caused by this pathogenic E. coli. So why is this E. coli a good pathogen? So see, it is a pathogen. We say it is bad, but it is a good pathogen. It means that in that category, it plays a very good role. Okay, so it is because it has the versatility in growth environment. So in any environment, it will grow. If you're putting it in the media, it will grow. If you're giving it in a food sample, it will grow. So wherever it is, even in the shit, even in the fecal samples, it will grow happily. Wherever it is, it will grow happily. So it is very versatile organism in the growth, towards growth in the environment. So it is easily transmissible. Next, its persistence in nature. So nobody is gonna inoculate it. It is naturally it is present in the environment. Next is the ability to adapt itself to the environmental factors such as pH, osmolarity, temperature, etc. So it can adapt. If the pH increases, it will adapt towards it. If the osmolarity is more or less, then it will adapt to it. If the temperature is high or low, it will adapt to it. So anywhere it can adapt and start growing. So it is a very versatile organism. So next is the ability to evolve into a more fit bacterium through genetic recombination via conjugation. That's why in many genetic studies we use E. coli for conjugation. Then whatever it is, even in the phage infection and multiplication of viruses, for everything we use E. coli. So E. coli is a very fit bacterium through genetic recombination. So this is how it became a pathogen also. It was a common saw. How it turned to a pathogen is because of the this genetic recombination. So what are the pathogens of E. coli causing diarrhea? So EPEC, that is enteropathogenic E. coli, enterotoxigenic E. coli, ETEC, EIEC, that is enteroinvasive E. coli, EAEC, enteroadherent E. coli, and EAEC, that is enteroaggregative EC. We say it as EAGGEC, okay, enteroaggregative. Next is the EHEC. This is the hero of our presentation now, enterohemorrhagic E. coli. So with certain sets of serogroups and virulence factors causing enteric and diarrheal diseases. So EHEC, we are going to see about this EHEC and certain serogroups. So in which serogroup it belongs to and which is pathogenic. So everything it deals with the next, the virulence factors and primer sequences. So primer sequences are not needed for you, but just the virulence factor you can see. The EHEC has two virulence factors. One is STX1 and STX2. So the word STX means Shiga toxin, Shiga-like toxin 1 and Shiga-like toxin 2. And EAE is entracing and then adhering gene. Then EPEC, BFPA, bundle fibers protein, and EAF. Then this aggregation is EAST. Okay, so these are all the probable primers with the virulence factors of this pathogenic E. coli group. So this is the general introduction, and this Shiga toxin producing E. coli, that is STEC. Next is the enterohemorrhagic E. coli, EHEC, and E. coli O157. That is O, it represents the serogroup O157, which is a very pathogenic strain which causes HUS and HC, that is hemorrhagic colitis and hemorrhagic uremic syndrome. Okay, these are often used interchangeably with when discussed as threats in threats to food establishment. So when we are discussing the threats of food, the food pathogens, these three organisms, that is STEC, EHEC, and E. coli O157, are interchangeably discussed. So most of the people think all these are same, and the question may be, the answer may be yes or no, because they may be same or they may be different also. It is all very well studied still. Next, the STEC strains are categorized by two characteristics. Okay, this Shiga toxin producing E. coli, STEC, it is categorized by two characteristics. One is their ability to produce the virotoxins. Okay, it will be toxic to the viro cell lines, that is virotoxins, and Shiga toxins, STX, that is Shiga-like toxins. Shiga means it represents the Shigella, okay, which causes dysentery. Okay, then so the toxin will be similar to the toxin which is produced by Shigella species, which cause the dysentery. Then their ability to adhere to cell membranes. Then while there are several STX variants, the Shiga toxin variants, two have been associated as the positive agents of human illness. So those two variants are STX1 and STX2. So these two are very important. And EHEC is a group of pathogens which has recently, in the year from the year 1980s, ever since the outbreak was in 1982, the first outbreak of EHEC. So from that year onwards, it gained its significance in foodborne gastroenteritis. So it is estimated that the pathogen is responsible for so many acute illness, of which 3,890 cases are hemolytic and uremic syndrome out of this much. Then 270 cases show permanent and end-stage renal diseases. So it will start with normal, usual diarrhea and vomiting like that, like how a foodborne illness will be. But it will end in a serious condition of renal disease and finally towards death also, as the renal failure will happen. Okay. Then the EHEC has been associated with two major diseases. As I told before, one is HC and another one is HUS, that is hemorrhagic colitis, which is an inflammation in colon leading to abdominal cramps, bloody diarrhea, and fever associated with HUS. The second one is hemolytic uremic syndrome, which is a disease resulting in acute renal failure, then the microangiopathic hemolytic anemia and thrombocytopenia. So these are the trade-off signs of this hemolytic uremic syndrome, HUS. So first it will be thrombocytopenia, next hemolytic anemia, finally it will be renal failure also. Okay, the endothelial damage in the kidney happens as the organism is able to adhere to the cells. It will adhere to the cells and it has the ability of heme agglutination. It can adhere to the RBC cells and it can make clots also. Okay, it can make clots and it makes lesions. It causes lesions on the kidneys, so that finally due to this toxin production the kidney loses its function. So there will be occultation of renal vessels. Okay, occulation means there will be bleeding inside the renal vessels. So how do these E. coli become such killers? So as I told before, E. coli was a silent commensal. It was very silent. And how it became a killer, such killer? It has been postulated that E. coli O157H7, that is the strain name, which is very important in the group EHEC, it became infected by a lysogenic bacteriophage. So we all have studied the lytic and lysogenic cycle and all, the viral bacteriophage life cycle. So how a bacteriophage adheres to a bacterium, it inserts its genetic material inside the bacteria, then it replicates inside and finally it lyses the cell and come out, so that it will carry the gene of bacteria as well as it will have the its own genetic material also. Okay, so this is how it happens. Once a lysogenic bacteriophage named 933 it adhered to a Shiga toxin producing Shigella species. Okay, to it infected the Shigella and it took the toxin producing genes STX1 and STX2 from the Shigella. So it holded that STX1 and STX2. So after holding it, it again went and infected the E. coli. So this horizontal gene transfer. So from this bacteriophage to from the Shigella species to the E. coli by a bacteriophage, that is 933 lysogenic bacteriophage. This recombination happened. So this STX producing genes, it entered to a E. coli and thus that E. coli become STX producing E. coli. So this EHEC emerged as a pathogen. Okay. So there are many serogroups like O26, O111, O55 associated with EHEC, and there are some non-O157 groups also, which doesn't belong to the O157 group, but it will cause the similar illness because of this STX1. Because once one lysogenic bacteriophage infected the Shigella and it did this horizontal gene transfer, and there have been years. No? After these years, from 1982, they studied this, and from that year onwards, from 82 onwards, from now, it has been almost 40 years. In this 40 years, many bacteria, this species, the toxin-producing genes would have transferred through this genetic recombination. So many pathogens like this may be emerged. We don't know. So the epidemiology worldwide. How is the epidemiology? How, where and all the outbreak took place? So all over the world, including underdeveloped, developing, and developed nations, after 1982, it has been captured in foods, slaughtering areas, and handlers. So you should remember the place where where the outbreak started: the food, slaughtering areas, and the handlers. Okay, next, the EHEC outbreaks, right from 1982 to 2002 were precisely monitored in the study of Rangel et al. So I've given few references here who all did the study in this EHEC outbreaks. So in Tanzania, then in Iran, in England, in Japan, China, and also in recent 104 strain outbreak in Germany, that is 2012. It is almost 10 years before. Next, what is the status in India? So, okay, being a developing nation, India tries to monitor the food safety to curb the food-associated health problems. In India, it is seriously, it is a very tough process of doing this healthy food and the fecal contamination free food, because there are more slums in India. So the number of slums is more in India and Africa. So these kind of foodborne infections are more in these places. By instituting the Food Safety and Standards Authority of India since 2006, and yet to take firm steps to regulate the unhygienic practices, thus reporting to several outbreaks in India. Okay, because there are many places which practice very unhygienic procedures in slaughtering, in packing foods, etc. So we, followed by this, we'll have a case study which will show you how the outbreak is happening, because how there are chances of outbreak in India. And the studies associated with O157 and non-O157 EHEC have been investigated by the Food and Water in Kolkata, then in Cochin also, in Arunachal Pradesh, and in Mangalore. And many studies from the clinical and environmental samples have been reported by Dr. Ghosh et al. 1991. And the occurrence of enterohemorrhagic E. coli in beef was carried out to determine the prevalence rate in Thrissur recently, that is 2014, 6 years before, and more others. So we'll enter into the case study in Vellore, the place where the case study was done. So this city is located 150 kilometers away from the seashore. That is, from Chennai, it is 2.5 hours travel. So it is 150 kilometers away. And it has slum areas practicing open defecation and improper sewage channels in few places. So the reports on diarrheal outbreaks in villages of Vellore and the incidents in Vellore city, these studies were done by the CMC, that is Christian Medical College Research Group, and these report says that there are outbreaks of this EHEC in Vellore also. So, but there are no reports on prevalence of E. coli in meat and meat-based fast foods. So this particular thing gave us a way to start with this case study, that is going for the meat and meat-based fast foods. So you can see this picture, see how unhygienic it is, the dumped garbage and sewage near the drinking water place. So here's the corporation water tap, and very close to it is the drainage, and it is dumped by the garbage. And there is a slaughterhouse near this also. I'll show the picture of this of that slaughterhouse also. So it is a very unhygienic place where very close to this place the meat is sold. So this is the unhygienic slaughterhouses with improper waste management in Vellore. So you can see there is no drainage system here, and the waste after cutting everything is the colon, whatever the waste from the slaughterhouse is thrown away as such near the slaughterhouse. And so the flies sit there and it take up the matter in the colon of these animals. Okay, definitely there will be our case studies hero, Mr. E. coli. So that E. coli will be entering the meat, and when this meat is not properly cooked, definitely that hero enters our body and it causes foodborne illness. So why this study? So in spite of several types of E. coli, the one causing HUS and HC, that is hemolytic uremic syndrome and hemorrhagic colitis, offers a red alert for high contamination in meat-based food and fast foods. So the distribution of such kind of pathogens in our environmental settings can be known by this study. So this is the reason for why we took this case study. Next is the E. coli, which remains as a good indicator because it is called as an indicator organism. I think you have studied in your subjects, in your core subjects also. Okay, so to check whether the water is potable or not, to check whether there is any fecal contamination, we detect E. coli. So if when E. coli is detected, there is a confirmation that the particular water or food sample have the fecal contamination. So it is a good indicator organism, and it is feasible to identify in the laboratory. So for detecting E. coli, you need not spend more, or you need not need very big the things, the laboratory chemicals or any equipments and all. Very simple agar and very simple media is enough to detect E. coli. So for these reasons, we have selected this case study. So we are entering into the study now. The study design is to isolate, to identify and characterize the E. coli strains possessing virulent genes and multi-drug resistant from meat and fast foods in and around the Vellore city. Hope you understood the case study's title, the design of the title. So we are going to isolate E. coli, we are going to identify the serotypes or whatever it is, and we are going to characterize whether it has any molecular pathogenicity, and we are going to check the antimicrobial resistance or sensitivity and around the city, the Vellore city. So all these things we are going to do it from the, we did it from the meat and fast food samples. Okay. So the first thing is isolation, how we isolated it. This picture illustrates the isolation shortly, that is from the meat and fast foods and the water present in that. And we carried in the transport media, we carried all these samples in the transport media, took it to the lab. It was streaked on to the McConkey, the differential media, which differentiates the lactose fermenting and non-lactose fermenting. So this E. coli comes under the group lactose fermenting. So the pink colonies were picked, and then it was subjected to the standard biochemical test for IMVIC for E. coli. Then it was confirmed in the EMB media, that is eosin methylene agar, methylene blue agar, where the E. coli gives a specific characteristic metallic sheen. See how the beautiful color is, the metallic sheen. It will be very shining colonies. It will glitter like a gold. Okay, so these colonies says that E. coli is present, and see, you can catch the laughter aloud, the laughter of E. coli in this isolation. Okay, next, the objectives: the collection of samples from the meat stalls and fast food centers, then the isolation of E. coli from the collected samples, and the microbiological and biochemical identification of the isolated strain. So the same thing I said with these pictures. So first, the collection of samples. So it was a random sampling method, and the sample number is 215. Excuse me. So the sample number is 215, and weekly once for a period of 18 months, we collected the samples, and the sampling sites are quite interesting. They are the meat shops. In the meat shops, we have sub sites also. The knife which they use for slaughtering. Okay, from the knife we took a swab. Okay, next the slaughter platform swab. The platform, the wooden platform or whatever it is where they chop the meat. So from the platform the swab. Then the hands of the butcher. The butcher, I don't know he washed his hands or not after going here and there. So the swab from the hands of the butcher. Then water which he used there, he just rinses his hand like this, he sprinkles on the meat. So he does all those unhygienic things over the in the meat shop. So that water was collected. Next, the meat itself, which we take it to home for cooking. Okay, so that meat, a piece of meat, then the processed foods from that meat, how the food is processed from the fast food shops. They just smoke it and they crush it and use it. I mean, they mince it and use it for hot dogs and whatever it is. The burgers thing, the cutlets, what they use. Anything. Okay, so the processed foods and the fast foods used with this, I mean the meat based foods, and some fish stall samples also. Same way, the fish cutting knife, the fish cutter, so everybody was swabbed. Next, the fast food centers. So these were the sampling sites and sub-sites. So this was the distribution of samples from spinach where we use it for burgers also, and we eat it, and some waters, and this water around the slaughterhouse, it may be drained to the agricultural field also, or it may be drained to the gardens where they grow the spinach also. So the spinach sample was also collected, 10 of the spinach samples and 6 smoked beef sample, and 12 veg rolls, and 5 salads, 5 pizzas, sandwiches, next hot dogs and chicken rolls, veg burgers, everything was in the sample. That is 215 samples holds all these things. Okay, next the water sample also, water, meat, hand swab, slaughter platform, knife, everything. So totally including the 15 samples from the fish stalls, the sample number is 215. So next is the E. coli from the samples. How the E. coli was caught from the sample collected. Okay, so the samples in transport media. Okay, we'll be carrying the transport media. The previous day itself, we'll prepare the transport media. We store it in the ice box along with the sterile swab in it. The transport media is buffered glucose broth. What we used is buffered glucose broth for transporting. Okay, so this buffered glucose broth media is taken to the meat shops, and with that we take this sterile swab and we swab the sampling sites as we shown before, and we put it again in the broth, close it tightly, hold it in the ice box again, and carry it to the lab within 2 hours. So within 2 hours, it will be taken to the lab and processed there. There, what we do is it will be enriched in the media. From that, we inoculate it directly to the McMonkey agar plates, and the next day we get the lactose fermenting and non-lactose fermenting. And for this NLF, that is non-lactose fermenting, it was collected for other foodborne bacteria. It may be some other foodborne bacteria also. Even Salmonella gives non-lactose fermenting colonies on McConkey, so it may be Salmonella also. But as we were focusing on only E. coli, we concentrated on the lactose fermenting colonies. So this lactose fermenting colonies were picked, and the standard biochemical tests were proceeded. Okay, so this standard biochemical tests are catalase, oxidase, TSI agar, that is triple sugar 9 agar, mannitol motility, and the IMVIC, that is indole correction, then the MRVP and citrate. So after this standard biochemical assay, we move on to the EMB plates, as I shown before, the metallic sheen colonies. So those, after confirming the metallic sheen colonies in eosin methylene blue agar, it was stored in a slant, or it was stored in the deep freezer, or the room temperature itself in the slant or slab. The glycerol stock was made and labeled properly. All isolates were given labels. The numbers, the codes were given, and it was hold it for the future work. Future work in the sense, whatever we did after that, the serotyping, the molecular studies, the salt aggregation, everything. Okay. So this is the flowchart of the samples, how we got the E. coli from the samples. So the distribution of E. coli, out of this 215 samples, this distribution, the distribution was from the meat shops, 100 samples were collected. Out of 100, 11 isolated, 11 samples holded E. coli. Okay. Next, the fast food samples. So from the fast food samples, 100 samples, 19 were caught. Okay, 19 E. coli was isolated from 100 fast food samples. Next is the fish sample. From 15 isolates, 6 were taken. 6 were isolated. 6 number of E. coli was isolated. So the totally out of 215, 36 isolates was picked. Okay, so there are some other isolates also which were not E. coli. That was about 25 isolates. So we hold that for other assays. So the E. coli isolates were numbered with the code that is PV from 1 to 36. So PV1 to PV36. This 36 strains were named. So next is the biochemical characterization of the gram-negative rods and how it exhibited lactose fermenting colonies on McConkey and the metallic sheen colonies on EMB. So the triple sugar ionograph test was A by A gas positive and H2S negative. So this A by A indicates acid sand and alkaline bud. Sorry, acid sand and acid bud. Okay, for alkaline you give K. Okay, so this is A by A is the whole sand, sand and bud, the whole tube will be acidic. So it will be yellow in color. And there won't be any H2S production, so there won't be any black color precipitate in the junction of bud and sand. And there will be gas production, which is indicated by the agar breakage or agar jumps. The agar would have jumped inside the tube itself. Okay, so that will be very colorful and beautiful to see this triple sugar ionograph test. And next is the indole production. It's a very beautiful color production. It is done in the peptone water. So in peptone water, you inoculate the E. coli. The next day, you give few drops of the reagent for indole production. So after giving the coax reagent, it gives a cherry red color compound. So that shows the positive for indole production. Next is the methyl red and Ovis Prosker. So for any organism, if the methyl red is positive, the Ovis Prosker will be negative. So if this is positive, that will be negative. So always this MR and VP will be vice versa. Next, the citrate utilization. So the color of the media will remain green only because E. coli is citrate negative. So, but Klebsiella, for this, one thing I want to say that the same results will come for Klebsiella also. Okay, the triple sugar ionograph, indole, methyl red, and Ovis Prosker. But it differs in the citrate utilization because Shigella, this E. coli is citrate negative and Klebsiella is citrate positive. So only with that we differentiate what is E. coli and which is E. coli and which is Klebsiella. But those who have experience, they can find out with the colony morphology itself because Klebsiella have mucoid colonies. It will be about shining and glossy colonies, and it will be very mucoid, moving here and there like that. Okay, and it will be very sticky also. It will be very shabby to see actually. And the mannitol motility test. Surprisingly, this 4 isolates among the 36 were non-motile, and the catalase test was positive and the oxidase was negative. So this is the biochemical test result. Next is the discussion. It appears that the sample material has fecal contamination indicated by the organism, and as they are distributed from the different places, these strains need further validation and characterization. So what are those further validation and characterization? So the 36 isolates were studied for further information. So we are moving on to the next section of characterization. So first thing is isolation, that is over. Next we are moving to the characterization. The objective of this chapter is to validate the isolates, that is PV1 and 36, using the designed primer. A primer was designed for this bacterial food pathogen. Only for Shigella, Salmonella, and E. coli, this primer was designed, and that primer we used to validate the E. coli. Okay. Next, to identify phylogenetic groups they belong to, and next is the using specific markers we found the phylogenetic groups. Next is the validation of E. coli. So in infectious diseases, prior to treatment, identification plays a crucial role, because before identifying what infection it is, so it may be about Corynebacterium infection, or it may be Shigella infection, it may be Salmonella infection. So what we don't know which organism caused the food illness. So before the prior treatment itself, the patient goes to the end stage, that is the renal failure in case of EHEC, not the very small or simple infections. I'm talking about EHEC. Okay. Next is the PCR. So it is effective process of species identification in terms of both rapidity and specificity. So very specific gene we can identify by the PCR, and rapidly also. So in just in 1.5 hours or 1 hour 20 minutes, the gene will be identified. Next, we attempted to design a single common primer for Shigellosis using bioinformatic tools. Okay, so these validations were done. The validation with a designed primer. This primer was used. The primer is called SHRKG, RKAG. That is, it was designed by Ratnakumar and Dr. Ghosh. Dr. Ratnakumar and Dr. Ghosh for Shigella. So it gained the name SHRKAG. So this primer we used for the validation. This is the sequence of the primer and the conditions what we used I am given here. And next is the phylogenetic grouping. So which phylogenetic group does the isolate belong to? So one is A, B1, B2 or D. So where A and B1 are categorized as commensal and B2 extra-intestinal pathogenic strains, and D to a lesser extent of intestinal pathogen. So B2 or D will be pathogens. If it is A and B1, it will be a commensal. So where we can differentiate the hero and the villain. So certain DNA fragments might be specific phylogenetic markers. By the literature reference, by the support of the literatures, we found the virulent gene signatures. So those signatures are THUA, YJA, and TSPE4 for the specificity. The specificity is THUA1A is for heme transport protein for EHEC. So if that protein is present, definitely it will be a pathogen, and it falls under the category of B2 phylogenetic plays. Okay, next, YJA is an initially identified gene by E. coli K12 strain. The function is unknown about that. And TSP is an anonymous DNA fragment. So with this, we move on. There is a decision tree with the support of literatures we studied to determine the phylogenetic groups of E. coli. So this THUA, if it is positive, if this gene is present in the sample, it belongs to B2 or D, which means it is a pathogen. And if it belongs to B1 or A, it will become a commensal. And when it is B2 or D, we go for YJA. YJA is to differentiate whether it is B2 or D. Okay, so when the YJA gene is present inside the E. coli, it shows that it belongs to the B2 group, which is an intestinal pathogenic group. And D, if it is negative, it shows that it belongs to the group D, phylogenetic group D, which is also a pathogen, but to a lesser extent. And if this CHU A gene is negative, it may belong to B1 or A, and to differentiate whether it is B1 or A, we go on to the other primer TSP-E4. If it is this TSP-E4 is present, it belongs to B1, and if it is not present, it belongs to A. So this is the phylogenetic tree. So primers used for this phylogenetic grouping is given as it was in the literature. We used it, and so this is the validation. So it this validates the E. coli. It validates the isolates as E. coli with amplicon size 153. So if this designed primer shows that if it is 153 base pairs, it shows that it is E. coli. So all this PB 1, 2, 3, 4, 5, 6, and 7 are shown here. We did this for all 36 isolates, but only 7 are shown here, which were all E. coli. It was validated as E. coli. Next, coming on to the CHUA gene. So it was present and it belonged to the group B and group B2. So we'll come to the discussion of this phylogenetic grouping. The identification of phylogenetic groups revealed that the isolated strains belong to phylogenetic group B2, representing the extraintestinal pathogens as they were qualified for the genes CHU A and YJA. So yes, it is a pathogen. But how it became a pathogen? What are all the virulent gene signatures in it? So we'll move on to the detection of virulent gene factors. So this objective is to reveal the pathogenicity in the E. coli isolates and molecular characterization. Next is the detection of virulent genes present in the isolates, the so-called pathogens. So the detection of virulence factors. A group of pathogenic markers were used for PCR analysis of isolates. So those group of pathogenic markers are STX1, STX2, which are very important. And next is the EAE, that is effacing and adhering gene, and EAF, that is adherence factor, and EAGG, the entero-aggregative gene, and BFPA, the bundle forming pilus A protein. Okay, so these pathogenic markers were used for the PCR analysis. And the results, it showed that STX1 positive and negative for STX2. STX2, EAE, EAF, EAGG, and BFPA. So for everything except STX1, it was negative. Okay. So this showed that isolates possessed STX1 gene, that is Shiga-like toxin producing gene 1. Okay, so the molecular discussion confirmed, the molecular characterization confirmed that 95% of the strains possessed STX1 gene encoding Shiga toxin causing HUS, that is hemorrhagic uremic syndrome, a category of E. coli called EHEC. See how finally we have got that EHEC, the most emerging pathogen. So whether this strain belonged to O157, which is a very deadly strain and which caused the outbreaks in Germany, or not. So we'll find out that. For finding that, we went on to the serogrouping, because we need to know the serogroup of the E. coli isolated. So this serogrouping was done, and after that, we did the cell surface hydrophobicity by salt aggregation test and hemagglutination assay. Then to analyze the antibiogram, because the resistance and sensitivity of the drugs have to be known. Okay, so these are the objectives of the study. And to continue, the serogrouping of E. coli was done. Next, antibiogram was done. For antibiogram, we used the proper Kirby-Bayer antibiotic assay and the major fluoroquinolones which are used for treating the E. coli infections. So those 10 antibiotics were used, that is ampicillin, chloramphenicol, ciprofloxacin, lomifloxacin, nalidixic acid, norfloxacin, ofloxacin, nitrofurantoin, streptomycin, and tetracycline. Okay, these were used from high media. And the salt aggregation test, SAT we call it, salt aggregation, was performed using molar concentrations of ammonium sulfate solution, and the cell suspension was made using PBS, and we used it for the salt aggregation test. The same similar way we did the heme agglutination assay also, with 20 ml of blood sample and 20 ml of bacterial suspension, and in a moist chamber it was done. So this heme agglutination and salt aggregation gave us few important points, and we'll see all together in the end of this discussion. And the distribution of various serotypes of E. coli. See what all serotypes we found: O8, O19, O22, O41, O60, O101, O116, and O147. Okay, so there are some virogenic strains also in this. So this serogrouping was done in Kasoli, the National Institute of this. Okay. Then the antimicrobial resistant pattern of E. coli isolates. So out of this 10 fluoroquinolone drugs, what was the resistant pattern of E. coli isolates? You can see very clearly in this picture that it was this PV'23, 31, and 33. These strains were completely resistant to all the antibiotics we used. So almost all the isolates were found to be drug resistant, at least for 4, to the least of 4, and the maximum of 10, it was resistent. So this graph represents the antimicrobial resistance of E. coli, and the ampicillin, for 19%, it was resistant, and chloramphenicol, and the 100% resistant was given to ciprofloxacin, lomifloxacin, norfloxacin, and ofloxacin. So the surface hydrophobicity, that is the SAT test, salt aggregation. 17 isolates were found positive in salt aggregation, representing the interaction between host and pathogen contributed by fimbrial appendages and non-fimbrial appendages. So it represents the aggregation, the attachment. The first category of, character of a good pathogen is the pathogen should be able to attach or adhere to the intestinal walls or wherever it is going to infect. Okay, the target area, it should first adhere. So only after adhering, it can colonize, it can grow, it can produce toxins. So this surface hydrophobicity is done for the fimbrial appendages and non-fimbrial appendages also. Next, 6 isolates were positive for HA, that is agglutination. It was able to aggregate aggregate the RBC cells. So it will attach to the blood cells, and it will do the clots or clumping of the blood. Okay, so this discussed the HA and SAT, that is the heme agglutination and salt aggregation, represent a new class of colonization promoting antigens and additions. Next, the pathogenicity is not only by the possession of virulent genes, but also by the resistance to fluoroquinolone. So definitely this drug resistance shows that it is a pathogen, because even after giving the drugs, if it is not responding to the drugs, definitely it is a pathogen because it creates a problem. So next is the summary of this case study. There are 36 strains, out of which was 4 non-motile from 215 samples of meat and fast foods were identified as E. coli by biochemical methods and validated using and validated using this primers CHUA, YJA, and they were identified as extra-intestinal pathogenic group, that is B2. Okay. Then after that, what we did, we found that those are MDR strains, that is multi-drug resistant strains belonging to various phylogenetic serogroups also. Next, the 17 isolates were found to be surface hydrophobic, that it was able to adhere to the it was able to adhere to the the target place, the target site, and 6 were able to agglutinate the erythrocytes also. Next, the detection of prevalence genes by PCR revealed 25% of the strains possessed STX1. Okay, so STX1 is sugar-like toxin producing gene. So this gene encoding to sugar toxin, which may be the cause for HUS, that is hemolytic uremic syndrome. Next is on serotyping the isolates, the distribution of various virogenic serogroups were found, and it was identified as virogenic serogroups. And the conclusion, this case study concluded that the detection of virogenic serogroups of STX1 harboring E. coli with multi-drug resistant in fast foods and meat shops alert us to create awareness among public about the unhygienic practices, because I've shown the pictures of the unhygienic slaughterhouse practices, and this creates awareness among the public about the unhygienic practices followed in the fast food centers and retail meat shops. So the improper disposal of fecal and antibiotic waste, which is the cause for such contaminations, should be taken care. So this study also insists the need of health education and hygienic practice for the food handlers. So it is a red alert for the people in Vellore. So only in Vellore the case study was done, so it is red alert for Vellore. If somewhere else the case study is done, then definitely there will also be a red alert, I guess, because we know what condition prevails in India, the hygiene, how it is in India, and the challenges in detection. So what are the challenges of EHAC? Now the threat is over. So overall, we have seen the threat. What is the threat in the environment? Threat of EHEC. Next is the challenge. It comes in the detection alone. Okay. So in June 2012, the US Department of Agriculture, Food Safety and Inspection Service, that is shortly called as USDA FSIS, so they implemented requirements for the expansion of STEC, that is sugar toxin producing E. coli, in testing, in testing in raw beef trimmings, because they use more raw beef trimmings and all, not in our country. In abroad, they, foreign countries use mostly uncooked meat. Okay, they just smoke it, they just trim it and use it in the salads. But we usually cook properly. But then, even though there are some improper, the handling, because of the handling and unhygienic practices, there are outbreaks in our country. So targeting the 6 additional serogroups which are connected to this EHEC, STEC, that is O26, O45, 103, 111, and 121 and 145. So made by a new reference standard for analysis. So with this, they made a new reference standard for analysis. So when these serogroups are present, definitely they say that it is STEC or EHEC. Then the creation of a complete polymerized chain reaction solution requires the skill of highly trained analyst, because if the trainer is not proper, and if he's not experienced, or if he's not handling the samples properly, definitely there will also be a outbreak. And additional testing is required to confirm the presence of these virulence factors, STX, and addition genes, so which are used to adhere, okay, EAE, effacing and attaching genes. Next, the variants of STEC. There are some variants in STEC strains which are closely related and can often prove that it is very difficult to detect in the environment. So this supplementation of media and antibiotics can be used to select the O157, which is the more pathogenic and the strain which gave an outbreak in the group which belongs to EHEC. But the recommended antibiotic for aiding in the selection of O157, the antibiotics is novobiocin, and it can completely or severely inhibit the growth of O157 strains. So isolation in the media, we were advised to use the novobiocin, which will restrict the growth of other E. coli and allow only O157 to grow, so that the O157 strain can be easily caught in the samples. Next is the molecular-based detection methods, which are traditionally done, and for a choice of O157 would need to be designed to detect genetic markers for multiple targets. So only for that specific gene. If you are going for one gene, and if that particular gene is not present, we may mistake that it is not a pathogen and we leave it as such. But that shouldn't happen. So it should be a genetic marker for multiple targets. So for multiple genes, it should be a marker. Next is the isolation of serogroups using antibody-coated beads. It's a technique-sensitive and cross-reactivity test. Okay, these isolation techniques have to be done. Next, the media used to isolate and the methods which require for skill. All these methods require the skilled analyst, the person who works in the lab or the researcher or anybody. The labor is a labor intensive and time consuming process. So for everything, we can't do it in 1 hour or 2 hours. It needs it is a time consuming process, and it is labor intensive process also. Every day you should follow up the results. So a strong need existed for improving the scientific approaches and expanding into new methods of detection and differentiation. So definitely there should be new methods of detection and the differentiation of the serogroups and STEC groups, non-STEC groups, EHEC groups, etc. So all these became a challenge. So there should be some improvements in detection also. And what are those improvements? Some ready-made kits have to be made, both in liquid or pellet form. The ready-made kits have to be made, and assays were designed to detect multiple target gene sequences, looking for both virulence and adhesion factors or the specific serotype. So like that, some assays should be made. Then the multiplex kits were developed and validated from multiple method developers, allowing laboratories to seamlessly integrate this analysis into the current system. So the detection remains a great challenge. So what will be the next step in the detection? The end goal of many in the food industry is a method designed to detect the top 7 serogroups that contain both virulent and adhesion factors in single, easy to use and affordable test kits. So we can catch all this top 7 serogroups in one test only. So in that way, it has to be designed. The method has to be designed. So this is the next step in detection. Thank you so much for the opportunity.
It was wonderful, Ashok. We truly appreciate you and your time spent by you helping us and learning new things in this wonderful occasion. We truly appreciate it, ma'am.
Thank you so much. Thanks for the opportunity. Thank you.
Now, all the participants kindly join your respective links for the presentation. And thank you so much, ma'am, again.